So what causes cancer? It's known that Japanese ladies have low incidence of breast cancer as long as they live in Japan. But if they immigrate and come to the United States, now the incidence of breast cancer in Japanese ladies who immigrated to the States does not differ much from the incidence of breast cancer in American ladies born and live in the US. Same with prostate cancer. It has low incidence in Japanese men as long as they are in Japan, but if they travel to the United States and they stay in the United States, they get higher rates of prostate cancer similar to American men. And before you think to immigrate to Japan, let me tell you that Japan has the highest incidence of gastric cancer in the world. So what causes cancer then? It's known that smoking causes lung cancer. Can you explain to me this picture? A lady celebrating her 100th birthday with a cigarette. Probably she has been smoking for maybe 80 years or more. So what causes cancer? Is it something in the genetics, or is it something in the environment? Actually, the answer is it's both. So it's nature versus nurture. So if you're going to be dumb, like the guy in this picture kidnapping the bear's baby, you'd better be tough. So if you're going to be dumb with your life and do all the unhealthy, risky behaviors, then you'd better have the good set of genes, because you're going to need these good genes to protect against all the bad behaviors that you might do. So cancer is abnormal cellular proliferation, abnormal cellular proliferation. And as I told you before, many genes and many proteins are involved in the process of cellular proliferation. Starting from this protein that is known as the growth factor. And they are not one protein, they are multiple proteins, known collectively as protein growth factors. And protein growth factors bind to specific receptors at the cell membrane. And again, these receptors are nothing but protein molecules. And this receptor protein is linked to a signal transduction pathway made of multiple protein kinases. And these are like the molecular on-off switches that pass the message down to the nucleus. And when the message reaches the nucleus, the DNA gets the message and starts to replicate to start the process of cell proliferation. So any problem with any one of these proteins, and we can use the words proteins and genes interchangeably because a single gene codes for a single protein. The gene codes for a single protein. So any mutation in these genes that code for these proteins that regulate the cell cycle can pass incorrect message to the nucleus: divide, divide, divide. And therefore, the cell will start dividing without having a real stimulation or a real need to divide. And this is the abnormal cell proliferation or cancer. So let me talk more about the cell cycle. Normally, our cells are not in a continuous stage of division, except some tissues like the bone marrow. The bone marrow cells are very, very active. They are in a continuous stage of division. They keep dividing like all the time. But most of the cells, they exist in the resting phase, which is referred to as G0 phase. And if a cell in the resting phase, or whenever a cell in the resting phase receives this protein growth factor, the stimulus to divide, it starts to accumulate nutrients, and this phase is described as the G1 or growth 1 phase. The cell is getting bigger because it's collecting nutrients, because the cell knows that soon it's going to be 2 cells. Then the second phase occurs in which the DNA replicates. And this second phase is known as the S phase. So G1 followed by the S phase, where DNA replication occurs. And this DNA replication phase is followed by another growth phase, and the cell will keep accumulating nutrients because it's going to be 2 cells. After the G2 phase, the actual mitosis starts, and mitosis ends by cytokinesis, where the single cell actually divides into 2 separate cells. So this is the cell cycle, which usually takes within 1 hour. And look what happens during this 1 hour. There are 3 checkpoints. The cell cycle is interrupted. Actually, it is stopped by certain genes or proteins, and everything is checked because the body highly values the process of cell division. And the body knows that you don't want to mess up with this process of cell division. If you mess up with it, you might end up having cancer. And this is something, of course, very dangerous. So it resembles actually like car racing. In car racing, no car is allowed to start the race unless it is instructed to. You know, this guy who shoots in the air and give this loud sound so that the cars start racing. And in order for the cell to start dividing, the cell has to be stimulated by protein growth factors, and they are known collectively as proto-oncogenes. So proto-oncogenes are the genes that code for proteins that initiate and maintain cell division. So proto-oncogenes are actually good genes. Like without proto-oncogenes, we wouldn't be able to repair the wounds. Without proto-oncogenes, we wouldn't be able to grow. So proto-oncogenes are very good. They are normal, they are physiologic because they initiate and maintain the cell cycle, like the guy who gives the signal for the car to start racing. And at one point in car racing, you notice that the car pulls over to the side and gets checked. And these are the checkpoints. So the checkpoints are like the booth or something where the car is checked. And what stops the cell cycle? Actually, what stops the cell cycle is this guy here with the stop sign. And this guy is another gene called the tumor suppressor genes. So tumor suppressor genes, they stop the cell activity, they stop the cell proliferation, they stop the cell cycle so that the cell can be checked. Is everything okay? Are you allowed to continue the race? Are you allowed to continue cell division? Exactly in a car racing. You notice that the car pulls over and a lot of people come around, maybe changing the tires, maybe checking the engine. We just want to check that you are safe and you can continue the race safely. And when the cell is checked, what if a defect or a problem is detected? Then they are going to send for this mechanics. Then they are going to ask the help of certain genes or proteins that are known as repair enzymes. And these repair enzymes fix the DNA, and the DNA is nothing but millions of genes. So whatever the problem is, some repair enzymes can take care of that. And once the car is ready to continue the race, then they allow the car to continue the race, and the cell continues the steps of the cell cycle until the next checkpoint, and the same process happens again. The tumor suppressor gene will stop the cell. Stop. You need to be fixed. If everything is okay, you can continue. If something needs to take care of, then we're going to send to this mechanic and the repair enzyme guy. The question is, what if the repair enzymes fail to fix the problem? In this case, the immune system will interfere and will instruct this cell to die by apoptosis. Not always the cell responds, because the cell can be so malignant that it does not respond to these instructions. But again, if everything is okay, the cell will continue till the third checkpoint. And again, the same thing happened. Tumor suppressor gene stops the cell, and the repair enzymes, if needed, are asked to fix the problem until finally this cell continues and divides into two cells at the end of the race or at the end of the cell cycle. So we can identify three major categories of genes. The proto-oncogenes, all of them are good genes as long as they do not have mutations. Proto-oncogene is a good gene. It is the gene that initiates and maintains, they are the genes that initiate and maintain cell proliferation. And it's not just one gene, it's not even 10 genes, they are hundreds of proteins coded for by hundreds of genes. These are the proto-oncogenes. They are good as long as they are not mutated. Also, the tumor suppressor genes. Tumor suppressor genes, very good, very protective, because they interrupt the cell cycle. They stop the cell cycle in order for the cell to be checked, so that if there is any problem, you can take care of this problem. And the third category is the repair enzymes. This mechanic guy. If there is any problem, we're going to take care of it. So these are the three major categories of genes and the three major proteins that regulate the cell cycle. So what if you have mutation in these genes? Now we're going to talk about the genetic role or the genetic control of cell proliferation. So there are mainly two opposing forces. Proto-oncogenes initiate and maintain cell division. Tumor suppressor genes, stop. You need to be checked. It's like driving a car and you have the accelerator and you have the brakes. You have the accelerator and the brakes. So if you think of the proto-oncogenes, you can think of them as the car accelerators. So the accelerator of a car, good or bad, of course, it's good. Without the accelerator, the car wouldn't be able to move. But what if this accelerator is stuck in the on position? Then the car will be self-sufficient in driving. Now the car is going to drive itself because the accelerator is stuck in the on position, so it's no longer you who controls the car. It's the car that is driving itself. So proto-oncogenes are like normally functioning accelerator, whereas mutation of a proto-oncogene, and when a proto-oncogene is mutated, we call it oncogene. So proto-oncogene is the normal good gene. Oncogene is the abnormal mutant gene. Oncogene is like what? Accelerator stuck in the on position. Oncogenes will result in what? Will result in the cell will be self-sufficient in growth. Self-sufficient in growth. The cell is not going to wait for a signal to start division, and the cell will start dividing and keep dividing on its own. That's the accelerator. The tumor suppressor genes, think of them as the brakes. So even with an accelerator stuck in the on position, maybe, as long as you have very good brakes, still you can control the car or have some control over the car. So tumor suppressor genes are the brakes, very, very helpful. You do not want to drive a car without brakes. And we have the hand brakes and we have the regular brakes. But what if the brakes were not working? What if the brakes were not working? So you're driving and you see a red light and you press the brakes and the car does not want to stop because the brakes are not working. So if you have a mutation in the tumor suppressor genes, the cell will be insensitive to anti-growth factors. So we have growth factors that initiate and keep cell proliferation, and we have anti-growth factor that stop and inhibit and slow down the cell proliferation. So what stops, what interrupts the cell cycle? They are the tumor suppressor genes. But if you have non-functioning, or if you have mutant tumor suppressor genes, the cell will be insensitive to anti-growth signals. An example of a proto-oncogene is the RAS. An example of the tumor suppressor gene is the p53 gene. The third category of genes are the repair enzymes, aka caretaker genes. Caretaker genes. So if you have a car problem, you go to the mechanic. If you have a computer problem, you go to whatever, the computer engineer or whatever. Do you ask a plumber to come and fix your computer? Of course not. What if you ask a plumber to come and fix your computer? The problem is going to get worse and worse and worse. So caretaker genes are like BRCA1 and BRCA2, BRCA1 and BRCA2. What are these? These are useful repair DNA genes. They repair the problems. But what if BRCA1 and BRCA2 themselves have mutations? Then if you ask them to fix a problem that happens during breast cell proliferation, they are going to make it worse. And therefore, for mutations of BRCA1 and or BRCA2 increases the risk for breast cancer and also ovarian cancer. And this is the genetic control theory of mutation of cancer, or this is the role of genetics in cancer. So when sufficient mutations, we also call mutations hits, have occurred, and thank God it's not just one mutation that takes you all the way to full-blown cancer. You need somewhere between 4 to 7 mutations in the genes that regulate the cell cycle. And when sufficient number of mutations or hits has occurred, 4 to 7, in the genes involved in the cell proliferation or cell division, cancer develops. And this explains why cancer increases with age, because as we live longer, we get exposed to mutagens that induce gene mutations. We get exposed to ultraviolet light, radiation, we get exposed to chemicals, we get exposed to viruses, and some viruses are involved in genetic mutations. So that's the reason why, as we get older, we get more and more gene mutations, and when we reach this 4 to 7 critical number of mutations in the genes involved in cell proliferation, if you get 200 mutations in other genes, you don't develop cancer. But the genes involved in cell proliferation, and they are proto-oncogenes, tumor suppressor genes, caretaker genes. Proto-oncogenes, tumor suppressor genes, caretaker genes. I have just mentioned examples for these genes, but they are more than that. So that's the role of genetics. So let me use my whiteboard to summarize So we have the good genes, proto-oncogenes, and tumor suppressor genes. Proto-oncogenes, so let me first comment what's the physiology or the function. Proto-oncogenes, like the car accelerators, they initiate and maintain cell proliferation. Tumor suppressor genes, like the brakes, they stop the cell cycle in order to check for any problems. That's physiology. What can go wrong with these genes? You can get mutations. A mutation in a proto-oncogene, we are going to call it oncogene. So proto-oncogene is a good normal gene. Oncogene is a bad mutant gene. And mutation in a tumor suppressor gene, we just call it mutant tumor suppressor gene. So what's the result of this mutation? If you have oncogene mutation, you're going to have the cell will be self-sufficient in growth. The cell will be self-sufficient in growth. Whereas if you have a mutation in a tumor suppressor gene, the cell will be insensitive to anti-growth factors. It's like driving a car without brakes. So you see the red light, but your car is insensitive to the red light because you press the brake and the car does not stop. So that's the role of genetics. That's the role of genetics in cancer. Mutations. Is there a role for the environment? The answer is yes. And we call this role epigenetics because it's something that happens on top of the genes that you have. So epigenetics is the effect of some environmental factors. And these environmental factors will determine or will cause the genes mutations to be either silenced or expressed. So let me explain this again using the whiteboard. So let me get back again to the whiteboard. So normally our genes are nothing but segments on the DNA molecule. And the DNA molecule is a very long coiled double helix like this. So let me take just one strand of it. It's very coiled. It's highly coiled. Why? Because it's very long and we have to put it inside the nucleus. And a segment on the DNA that codes for a single protein is called a gene. So this red segment is a gene. This blue segment is a gene. This green segment is a gene. This purple segment is a gene, etc. So we have thousands of genes. In order for this gene to be expressed as a protein, the DNA molecule has to unfold or uncoil. So let's say you want to make protein out of this red gene. In order to make protein out of this red gene, this part of the DNA has to be uncoiled. So this red gene is exposed. When it is exposed, now you can utilize it to make a protein. So what keeps these coils in the DNA molecule? At this point, at each point of coiling here, we have methyl group. Methyl group. So if you increase methylation, you're gonna keep the coils and these genes will not be expressed. So we say that methylation leads to silencing of genes. Any gene, methylation is going to silence it. Methylation is going to decrease the chances that this gene will be expressed into proteins. So if you know that a newborn has a bad gene that predisposes the newborn for colon cancer later in life, does this mean that this person has no chance whatsoever and he's doomed to develop colon cancer? The answer is not necessarily true, because I know that you have this bad red gene, and I know this bad red gene is going to put you one step closer to colon cancer. But maybe if I provide really a lot of methylation, like vegetables are rich in methyl groups. So if I give you 5 servings of veggies every day, that's a lot of methylation. I'm going to keep your DNA molecule highly coiled, and most probably this bad gene will not be expressed, and it will not put you one step closer to cancer, because epigenetics or environmental factors prevented the bad gene from being expressed or from being read. So they are called epigenetics because they do not change your genes. You still have the bad gene, but epigenetics, or the role of environment, it will determine is your gene going to be silenced or expressed. So methylation always causes silencing of genes. So it's good to silence bad genes, but it's not good to silence good genes. Sometimes babies are born with super mutation, a mutation that makes them resistant to cancer, actually. So you do not want to do methylation for them. You want to do other chemical processes like acetylation, because acetylation helps unfold the coils. So this is the process of, or the role of environment, or the role of epigenetics. And I think this is enough for this segment.